Hello, everyone. Good evening, good afternoon, and good morning to everyone joining us from across the globe. My name is George Maliaras, and I'm from Cambridge NeuroWorks. On behalf of the organizers, I would like to thank you all for joining us today for this inaugural Cambridge NeuroWorks and PolyU pair joint seminar. We are thrilled to see such a strong turnout for this collaborative milestone, and we look very much forward to a session filled with groundbreaking insights. And to continue our introductions, I'll hand the floor over to my co-host, Professor Chen Yan, who is the director of PolyU Academy for Interdisciplinary Research.
Thank you very much, George. It's also my great pleasure to welcome all the people from no matter where you are from to this joint seminar. This is the first seminar of the series, and we are very happy to have very distinguished speakers today. They are Professor Edward Chang from University of California, San Francisco. And then we also have 4 pitching session speakers who are Ms. Lucy Joan, Mr. Jonathan Fisher from the Cambridge NeuroWorks, and also Professor Sonata Yeo and Professor Bolton Chow from PolyU. So first, we'd like to play the pre-recorded welcome message from Professor Sir John Austin, Prof. Vice Chancellor for Research at the University of Cambridge, please.
Hello and welcome from Cambridge in the UK. I'm John Aston, the Pro Vice Chancellor of Research at the University of Cambridge. I'm delighted to be giving the opening remarks for this important seminar collaboration between Cambridge NeuroWorks and the PolyU Academy for Interdisciplinary Research. Neurotechnology has the potential to tackle some of the most stubborn problems in global health, problems that are damaging billions of lives and livelihoods. Depression, dementia, stroke, chronic pain, Parkinson's disease, incontinence, OCD. Just imagine a world where no one needs to take time off work because of depression, where people recovering from stroke can get affordable remote therapy in their own home, where no one lives with chronic pain, or the fear of falling over, or the fear of epilepsy, where we can cure dementia before it occurs. Neuroscience and technology has the potential to solve these global ills, but only if we create a space where the brightest people with the boldest ideas are free to take risks, experiment, fail, learn from that failure, access real patients and the doctors looking after them, and then get business support to help them create companies to manufacture their innovations at scale and for an affordable price. That is the purpose behind our Cambridge NeuroWorks program. It is co-run by 9 partner organizations across the city, including Cambridge University, and it brings together experts in engineering, neuroscience, AI and data, clinical trials, and business, all there to support groups of funded fellows who have come to us from around the world to turn their bold neurotech ideas into reality. You're about to hear from 2 of these Cambridge NeuroWorks fellows, Lucy and John, who will tell you about their devices. The first, to help people with anxiety, stress and trauma and sleep disorder. The second, to support people with Parkinson's disease when their legs suddenly freeze up out of the blue. One of the patients supporting our program told us of the frustration of being unable to get off a train at your stop, simply because you can't get up and walk off. Both Lucy and John had unique and very personal inspirations that drove them into neurotechnology. Lucy was diagnosed with a brain tumor in 2014, aged just 26. John's father suffered from Parkinson's disease. However, we can't solve such daunting, complex health problems in isolation. We need to join forces, and that is why we're so excited about partnering with PAIR for these 3 online seminars. The first examining the clinical need for neurotechnology, the second looking at the technology itself, and the third exploring how to make neurotechnology profitable. PAIR's focus on addressing major societal challenges, on global collaboration, and on partnering with academia, business, industry, and governments feels like the perfect match for Cambridge NeuroWorks and Cambridge University. and we are delighted that these innovative and globally focused institutions are joining forces. I'm very much looking forward to the series. Today presents an opportunity to hear from both inspirational global leaders such as Professor Chang and young innovators keen to make their mark. But it's also about creating long-term partnerships and friendships across the world, and it's about creating a community determined to improve the lives of billions of people. We encourage you to ask questions in the panel discussions and afterwards, to get in touch with us through the PolyU Academy and here in Cambridge through the Cambridge NeuroWorks website. Once again, welcome and enjoy.
Thank you very much, Professor Austin, for your very inspiring message. Now we would like to play the pre-recorded message from Professor Chris Chow, Senior Vice President for Research and Innovation at the Hong Kong Polytechnic University.
Professor Sir John Aston, Professor Edward Zhang, distinguished colleagues, friends and partners. A very warm welcome to all of you joining us online for the launch of the Cambridge NeuroWorks and PolyU Pair Joint Seminar Series. We are delighted to launch this seminar series as a brand new platform that brings together the strengths of PolyU and the University of Cambridge. It reflects our shared commitment to deepening knowledge, fostering innovation, and tackling some of the most pressing challenges in health and technology. Allow me to briefly introduce the PolyU Academy for Interdisciplinary Research, PAIR. PAIR is home to 20 research units, each with its own focus, yet united by a common mission to tackle complex challenges through cross-disciplinary collaborations. Our work broadly falls under three key themes: good health and well-being, advanced technologies and manufacturing, and smart and sustainable cities. PolyU places strong emphasis on research impact, translating ideas into practical solutions that benefit society. We are also deeply committed to enhancing global visibility through knowledge exchange and international collaboration. The Cambridge NeuroWorks and PolyU Pair Joint Seminar Series will undoubtedly add significant value to these efforts. I would like to express my sincere appreciation to Professor George Malieras of the University of Cambridge, who also serves as a PAIR Senior Fellow, and Professor Cheng-Yan Chen, Director of PAIR, for their leadership and dedication in making this joint seminar series a reality. This series is dedicated to advancing knowledge and emerging technologies that address diseases of the brain and nervous system. PolyU Pair already possesses strong capabilities in this area, with several units actively engaged in health-related research, including smart aging and mental health. We are also setting up a new unit focusing on microbiology in the coming academic year to further our strength in this area. At the same time, our technology-focused unit, spanning artificial intelligence of things and intelligent wearables, are developing innovations readily applicable to healthcare. Building on PolyU's strengths in artificial intelligence and aligned health, these initiatives form a robust foundation for driving the next generation biomedical devices and health technologies. Last but not least, this series is designed not only for listening and learning, it is also for fostering interaction and driving innovation. Through researcher pitches from both sides, as well as the Connect Health Tech platform, we hope to create meaningful opportunities for collaboration among academia, patients, charities, and industry. Thank you very much, and I wish you all an inspiring and productive seminar series.
Thank you very much, Professor Chow, for your encouraging message. Now it's my distinguished honor to introduce our keynote speaker for today's seminar, Professor Edward Zhang. Professor CHANG is a world-renowned neurosurgeon and scientist from the University of California, San Francisco. His pioneering work in the mechanisms of higher-order human biological functions, specifically in speech, memory, and emotion, has revolutionized the field of neuroprosthetics. We are incredibly privileged to have him here to share his latest insight. Professor CHANG, the floor is yours.
Good morning, everyone. Thank you, Professor Maliaris and Professor Chen, for this really remarkable honor. I'm really appreciative of the opportunity to share some of my work with this exciting collaboration that you are building. And this lecture is going to cover some of the highlights of our work from the last 15 years in an attempt to discover the neural code of speech. What that refers to is the basis by which humans communicate, the biology of that electrical code in the mind, in the brain, and then how we've been able to leverage that for building what we call a speech neuroprosthesis or a speech brain machine interface, the world's first device that was designed to decode words and sentences from the human brain. So this quote in particular really motivated me early in my career from Stephen Hawking, who suffered from a severe form of ALS, which is a degenerative condition where patients will lose the ability to move over time. He was only able to communicate through very small, laborious twitches of muscles in his cheek, probably about one word every couple of minutes. And basically, this quote that he created, I think, says it all for many of our patients that have this kind of syndrome, which is a kind of locked-in syndrome. Although I cannot move and I have to speak through a computer, in my mind, I am free. And so, as a neurosurgeon and as a doctor who sees many patients with these devastating conditions, this has been one of our major goals, is to really figure out how we can create a communication portal to the mind. When I began our work, a lot of excellent work was being done in the brain-computer interface realm, trying to work on communication systems for moving a computer cursor, typing, texting, handwriting. These are some of the communication rates for people who are normally communicating, not people who are paralyzed or BCIs. Handwriting on order, we can get to about 20 words per minute, texting about 40, and a professional typist can get up to about 60, 70 words per minute. One of the reasons that we were really interested in speech is because the ability to transmit information from my brain to yours and your brain to mine is really unparalleled in the information transfer rate. So speech is a very special behavior that has enabled our civilization as social animals to create shared knowledge and connection. And so being able to do that at about 150 to 160 words per minute is what I'm doing right now and is a goal for what we're trying to restore with the speech neuroprosthesis. Over the last 15 years, we've made some fundamental discoveries about the part of the brain that controls the ability to shape the breath into words. So what we're doing when we're speaking is taking the breath, the air that comes from the lung, and shaping it with the resonances of the lips, the jaw, the tongue, the larynx, to create syllables and words. We're shaping air. That's essentially where speech comes from. And we've been able to discover over many years how does that process actually work. This first paper that we published over 10 years ago now described the map, the blueprint for the organization of different parts of the vocal tract when people are speaking. This paper described the control of pitch, the change in the pitch of the voice controlled by the human larynx. We discovered a new part of the human motor cortex that was important for the control of the larynx. And then we've described all of the movements actually for the tongue, the jaw, and more when people are speaking. In fact, we discovered that what is known as the motor cortex, in fact, actually has auditory activity. So not only is your auditory cortex, the hearing part of your brain, processing the words that I'm speaking, we discovered that the motor cortex of your brain is also encoding the sounds of speech. And we also showed that these areas are tightly integrated using feedback control. These discoveries have been the basis of a translational effort in my lab to understand how to harness that technology to create what is known as a speech BCI. So a brain-computer interface that can synthesize the activity of someone's brain and translate it into words. And this is just an article that was covering some of the work that we were doing over 10 years ago. Could a person who is paralyzed and unable to speak, like Stephen Hawking, use a brain implant to carry on a conversation? One of the first things that we did was to prototype the engineering, the translation of brain activity into words in epilepsy patients. These were folks that were undergoing surgical implantations of arrays in the brain or on the brain surface. You can see that there's a large 256 channel array over the brain surface. And one of our first projects was to decode the neural activity from that brain activity into a model of movement called kinematics. And that model of movement, of the vocal tract movements, is then translated into speech that you can hear. This demo was the first time that we were able to demonstrate that using modern recurrent neural networks, a kind of machine learning and AI, we could translate the brain activity into words. In this video, you're going to first see the brain activity that's then translated into a virtual simulation of the vocal tract. And then you will hear the sounds that are generated from these movements. You'll first hear what was synthesized by the computer, and then you will hear what the person actually said in the microphone. This was an exercise in people that were not paralyzed. These were people who had epilepsy participating in this research study as part of their clinical mapping.
The proof that you are seeking is not available in books. The proof that you are seeking is not available in books. The proof that you are seeking is not available in books. Shipbuilding is a most fascinating process. Shipbuilding is a most fascinating process. Shipbuilding is a most fascinating process.
So what you're hearing are some of the very best examples that we were able to synthesize directly from brain activity. A lot of this speech actually you would not be able to understand, in fact, but these couple of sentences and many more were actually intelligible. And to us, that was very exciting. It was the first time that we could take brain activity and synthesize speech that we could actually understand. So that was a very exciting thing. And so we took the results of that study and applied for approval from the FDA to create what is now known as the Bravo trial. It's a very small trial that we started to explore the use of electrocorticography. These are brain recordings from the surface of the brain for brain-computer interface, restoration of arm and voice. So restoring movement of arm and voice information. And this is in partnership with my colleague, Dr. Ganguly in neurology at UCSF. We've implanted subdural surface arrays on the brain. The reason we're doing surface array is that we don't want to do injury to the very precious cells and brain populations that we're trying to record from. We want to safely record from them from the brain surface. And this is an array that's covering both the arm and speech areas. The first paper that we published from this was this article in the New England Journal in 21. It's entitled Neuroprosthesis for Decoding Speech in a Paralyzed Person with Anarthria. This term anarthria refers to a medical condition where people have lost the ability to speak. And the first participant in our study was a man named Pancho, who had a stroke 15 years earlier. The way that he communicates is this stick attached to his hat. And with this, he can communicate about 3 to 5 words per minute, very slow. The reason he became paralyzed was because he had a stroke in an important part of the nervous system called the brainstem. The brainstem is the part of our nervous system that connects your brain to your spinal cord. So when people have a stroke or injury to the brainstem, they effectively have lost the ability to transmit commands to the muscles in the arms and legs and to speech centers for speaking. They become very severely paralyzed in the arms and legs, and lose the ability to speak. This is about 1/5 the size of what the brainstem is supposed to look like. And everything that's processing above this in the brain is still working normally. So what we did was we created a device with the approval of the FDA to actually record from the speech cortex in Pancho. And we connected it to a HDMI cable that can transmit his brain signals to a computer that would then translate those electrical activities into words. So here you can see the illustration where the brain signals are transmitted to a computer that extracts this signal processing on the data. Then it extracts when he's trying to speak. Number one, it's a detection step. And then it tries to classify what words he's trying to say. And then that is displayed on a computer screen. This is what the port looks like. So each one of those tiny gold dots corresponds to a wire that is wired and covering a specific position on Pancho's brain. And then we connected it to a port that's connected to an HDMI cable. So this is what we call percutaneous brain-computer interface. And now we are making one that is fully wireless. Hopefully in the next year, this will be available. But for the last 5 years, we've been able to de-risk this technology, improve the technology with this tiny little port that he lived with for 6 years. And the area of the brain that we're particularly interested in is this area called the motor cortex. So the motor cortex is shaded in blue. The array, initially 128 channels, 8 by 16 channels, was sampling from the speech areas, the hand area, and also the vocal areas for detecting speech and decoding speech. We started with a 50-word vocabulary. So basically a set of 50 words that we trained a model to decode individual words. And this is what is called a confusion matrix, comparing what we instructed him to say versus what was decoded or predicted by our computer algorithms. And you can see that overall it's generally good, but there were some words that were very confusable, like it and is. For example, they have very similar phonetic structure and were confusable. But overall, this is tapping into the vocal control commands from the brain. This video basically shows one of the demos that we connected with Pancho. We gave him a prompt, And then we decoded from his brain activity, and this was implanted to a device that was decoding his brain activity. The blinking dots on the screen corresponds to him, the algorithm detecting that he's trying to say something, and then it decoded what word he's trying to say. You can see here this HDMI port is transmitting the signals out from his brain. So blinking dots means word detection, and then it's assembling the sentence in context, and then uses autocorrect and language model in order to make it faster and more clear. Something that's really important for this technology is that it lasts a long time. We've been able to record from weeks, months, and years after implant. This is an example of activity of one particular electrode. When he's saying the word goodbye, we can see clear activity that is occurring within a second of saying goodbye. We took care of another participant named Anne. Anne had a similar incident about 18 years ago. She had a severe stroke in the brainstem. Here's a picture of Anne and her husband, Bill. And this is the part of her brain that you can see is missing here because of the severe stroke. And we implanted a 253 channel array for Anne that's covering the motor cortex as well for speech. And we found very high performance just several days after we started. So about 25 days after the surgery, after the healing process, we connected her to the machine. And within a week, we basically were able to achieve near 95 to 100% decoding accuracy on words. So double the number of sensors, electrode sensors, gave us a huge improvement actually in the accuracy. And so with this, we began an even more ambitious project. We wanted to decode the probability of text phonemes. We wanted to decode the brain activity into speech that was synthesized, and then also to decode the brain activity into avatar movements. So this is an example of the performance that we then saw. On average, it was about 80 words per minute, sometimes up to 120 words per minute. You can see that Anna actually had 2 HDMI cables. Here, we're not decoding single words, but actually full sentences. And this approach continues to improve because it's becoming more and more accurate and also much faster. We've been able to do this now with sub-second latency and follow-up papers. So this is a large vocabulary of 7,000 words. And then here's an example of a demonstration where we're able to translate the brain activity into speech that you can hear, in addition to controlling a digital avatar.
Wonderful. Give me a few minutes.
Okay. And because we're building an avatar, we went ahead and proceeded to try to decode facial gestures, for example, expressions. One of the things that we learned from working with people with severe paralysis, it's not just the words that are lost, but also the ability to convey meaning through expression. It's a really important part of our identity to be able to express in these kind of ways. And so because our patients were asking us to do this, we use the algorithm to control expression as well. So this is just a demonstration of Anne essentially doing facial expressions through this digital avatar that we created. Digital avatar can have literally millions of different forms, and you can tailor them to exactly what you want. These are the look that Anne wanted. And we recently able to discover a way to use new kinds of AI to do very low latency streaming brain to voice. So this is an example of streaming brain activity in live synthesis. A lot of the previous work that we did would take the whole sentence and use the context of the sentence to improve the accuracy. But here, this is a very low latency streaming interface.
I am sorry, but I have an appointment now. Let me tell you what I did.
Okay. A lot of people ask me, Dr. Chang, can this work for, people who are bilingual? And we did that as a study. So we went back in to Pancho, whose original language is Spanish, and he learned English. And we built a decoder, actually, that decode whether he's trying to speak English or Spanish based on the brain activity alone, and then uses the language model from either English or Spanish in order to predict his words. So we can decode from the statistics of the brain activity what language he's trying to speak. It's not the brain areas, it's the same part of the brain, but different electrical patterns for when he's trying to speak English or Spanish. And we could tap into those to decode one language or the other. So just to summarize here, in this Bravo trial at UCSF, we've been able to make a lot of progress across many applications of brain-computer interface. One that I started with the brain to text, the brain to avatar, brain to facial expressions, bilingual decoding, and then work from my colleague, Dr. Ganguly, has shown that you can actually control robotic arm, all just from the surface of the brain without having to put electrodes into the brain or more. So this is just work that is continuing to develop and continue to improve with time. In the future, we want systems that are going to be very high performance. For speech, we want them to be 80 words per minute or more with low error rates, easy to use, that's something that will work for not just days, months, but years and ideally decades across multiple indications. We want them to be safe so that people can have privacy of their brain data and personal data. And from a surgery perspective, we want fully implantable wireless systems that is safe and easy to implant and explant. That's one of our main projects right now, is to build a fully implantable, easy to implant kind of system. So I wanted to conclude with what my general thoughts are, which is that as we move into the next chapter of this very, very fascinating world of brain-computer interface, we'll continue to do research, but we also are thinking about how to make these systems work in practice. I think in the next 2 to 3 years, we will see the first systems that will be used for people as commercial applications, to see how widespread this technology can be used to help people. And I'm very looking forward to be part of that exciting journey. So Professor Maliaris and Professor Chen, thank you very much for the very, very special opportunity to participate as the inaugural lecturer for your collaboration.
Well, thank you very much, Professor Chang, for sharing your wonderful work. I'm just an engineer. I don't know much about the neurosurgeon, but looking at your work, I really feel your work is super important. It really illuminated this keynote presentation to all the audience in the world. Thank you so much. So I know that there must be a lot of questions that people want to ask, but before we do that, I'd like to move to our pitching session. So in this pitching session, we'll have the showcases and let on the several different type of directions, which being conducted at the both Cambridge NeuroWorks and also PolyU pair. So today we have 4 speakers, and first I would like to invite Ms. Lucy Joan from the Cambridge NeuroWorks to present her work. Lucy, please.
Thank you so much for having me today. As I was introduced earlier, I was always working in a medical device innovation, whether I was working in medical devices for people with Parkinson's or multiple sclerosis or paralysis. So my passion lies in supporting people with neurological conditions. However, when I was 26, I was diagnosed with brain tumor, which led to lots of complications with medication and brain surgery. So one of the key things that I learned in innovating in Parkinson's and lots of medical devices is that sustained physiological stress can lead to hyperventilation, autonomic dysregulations, and panic-like responses. So if I was mostly working in motor conditions, I started getting really fascinated about overall how the brain is interacting with our body and how it kind of affects our day-to-day life. So that's why we set up Larynx Neurotech, where we're looking into neurotechnology and regulations in human status. Unfortunately, there's lots of people who are struggling with regulation issues, whether it can go into mental disorders or physiological stress. And if we leave this, the higher risk of dementia, Parkinson's, or other chronic stress can be significantly impacted. The earlier we restore the balance, the better we protect brain health, function, and future potential. So we started looking into the stress loop of when someone is going into the trigger, what actually happens, our central nervous system starts reacting to it. And if it goes to the panic, it can cause the trauma, and there's a negative reinforced fear for the trigger. So we wanted to stop that cycle, and we were looking into how we can do that. We started looking into co-regulation, physiological synchrony, and sensory rhythmic entrainment, where people can synchronize their central nervous system to other stimuli. So whether you're hugging someone or if you're exposed to other stimuli, it can really regulate or synchronize your body. The nervous system responds to slow, predictable rhythm, but the other thing that we're also realizing is that if it's a faster, predictable rhythmic actions, it also responds to that external stimuli. So we are currently developing an ABLE-1. It is a device that is mimicking our central nervous system. It does have 3 different stimuli using visual, tactile, and auditory cues, depending on individuals. Some people may be more sensitive to one sensory input to the other, which is designed so that it provides a mimicking nervous system so that we can synchronize one state to the other. It couples together with an application, and this is where it becomes very important for us, where we connect physiological and psychological and also our living status. Some people may be feeling completely fine, but when the life hits you, it can cause a severe stress, and that can also cause further impact. So we're observing, detecting, and learning to train so that we can support individuals. We're currently carrying out lots of scientific validations. We are at a very early stage, but we're very much looking into learning more. So we are currently looking to literature reviews as well as early impact of our body when we provide auditory or tactile stimuli. And we are seeing some very interesting insights into individuals being able to stay regulated whilst they're going for a walk or going for a study or performing. So this is one of the key studies that we are working on. But at the same time, I really loved what we're discussing today about real life impact. We are seeing how people are using the device at the moment. And one key thing is that there hasn't been any negative effects reported, but really lots of people coming back to us with how they're using the device and how it impacted them depending on what they're trying to do, whether they're trying to go to sleep or try to perform. So that's ourselves. We are aiming to fill the gap to support lots of people around the world so that we can enable lots of people to live their fullest. Thank you very much.
Thank you very much, Lucy, for sharing your work. That looks wonderful. Now I would like to invite Jonathan Fisher, also from Cambridge NeuroWorks, to present his work. Jonathan, please.
Okay. Amazing. Thank you for this opportunity. My name is Jonathan Fisher, and I'm working on treatments for mobility within Parkinson's disease. So this all started for me with my father. When I was at university, he was diagnosed with Parkinson's, and I saw all of these problems with Dad. And I became so frustrated that during my design degree, I decided to invent a device to help him walk and improve some of his quality of life. And that initial moment has led me on this journey onto which I'm developing it now. And so when you think of Parkinson's, it's more than just tremors. There's actually 40 symptoms, of which some are motor and some are more cognitive and non-motor. One of the biggest burdens in Parkinson's, however, is mobility. In particular, it's falls related to poor mobility. 68% of people with Parkinson's fall annually, and you're 4 times more likely to fracture your hip. One of the biggest problems in Parkinson's is symptoms called freezing of gait, which robs you of your confidence and stops your legs from working. Even though we've had fantastic advances in Parkinson's treatments and medication, mobility in falls is still one of the biggest clinical challenges we're currently facing. Now, cueing is something that's really interesting, as Lucy just briefly spoke about, and it's emerging as a fantastic and scalable non-invasive treatment for improving mobility within Parkinson's. So what is cueing then? Ultimately, cueing is providing an external timing or a rhythm or a framework for movement within Parkinson's. They can be visual, they can be audio, such as click, or they can also be effectively vibration, touch. Now, when you have Parkinson's disease, your internal timing of the way you walk and your movement initiation, as in starting steps, are massively impaired. And specifically, as we can see on the left, what happens is the loss of dopaminergic neurons in the substantia nigra contribute to this. On the left-hand side is a standard person's brain, and on the right, someone with Parkinson's. And if you look at the bottom with the U, that loss of that black line, which is substantia nigra, is something that happens as the disease progresses. Now, the loss of these neurons leads to reduced ability to walk and can result in freezing of gait. Cueing helps overcome these symptoms, however, by providing you a rhythm to walk with. And so this video here is going to show what I'm talking about a bit more succinctly. As you can see, this individual is walking, and suddenly his legs stop working, and his legs become frozen to the floor, and this is freezing of gait. It totally robs you of your independence and your confidence, and it just destroys your mobility. I have seen people who cannot get off trains, they cannot go through doors, they cannot live their life. They literally cannot walk anymore. And so, one day researchers decided to put lines of tape on the floor, just lines of tape, nothing else. And by looking at these lines of tape, this gentleman could walk again without any medication. And then he played a rhythm, and I'm sure you can hear it, but it's this very simple beat. And just by following this rhythm, he overcame this freezing. And he could even turn like a soldier exactly to the rhythm of the beat. So it's an amazing way to overcome this freezing and get you moving again without needing to be totally reliant on your medication. Now, effectively, cueing is a non-invasive treatment for Parkinson's. And as I said before, it can be visual, audio, or touch and vibration. However, one of the most important things with cueing is that you have to do it at a rate that is personalized and at appropriate times. If you do too much cueing all the time, a lot of clinical studies are showing that it's not as efficient in the long run. And so I saw all of this data when I was at university, and I saw all of these methods, and I decided to come up with an idea that I thought would be quite novel and unique for my father. So Peter's my father's name, and I decided to name the invention after him. And what I made was a wearable device that discreetly plays that audio that you heard in real time in a personalized manner. Now, what you're seeing here is the initial prototype. The form of what we're doing has changed, but the core concept remains the same. And that is we are providing invisible and discreet cueing so that no one in public can hear this, and you remain your confidence and you lose that stigmatization using clinically validated cues that we are testing as part of the Cambridge NeuroWorks Fellowship. And currently, we've built our prototype again. We're testing it with people with Parkinson's in the clinic with our study that's being used through the University of Cambridge. And it's incredibly exciting. And I have the next year now to develop this technology even further through the support of the Cambridge NeuroWorks program. It's been a total privilege to have a quick snapshot and share what I'm doing today. And if you are interested, please do connect with me on LinkedIn, because it'd be fantastic to have you all on the journey. And just thank you again so much for the opportunity. It's really great.
Thank you very much, Jonathan. And your family issues really inspire your work. I think this is great work. Now, next I will invite Professor Sonata Yao, who is from PolyU Research Institute for Smart Aging, to present her work. Sonata, please.
Thank you so much for the introduction. So I'm Sonata from Department of Rehabilitation Sciences, also the core member of two research institute and also research center in Pair. So I'm talking about the physical exercise effect on brain health. So my work mainly focuses on animal model. We use animal model to understand how physical exercise promote brain health using different animal models like depression, diabetes, and autism, so that we can identify some biomarker for translational research that can optimize the exercise protocol in different clinical patients. And also by understanding the underlying mechanism, we can identify some therapeutic drug target for discovering the exercise mimetic drugs treatment for different neurological disorders. So how exercise promote brain health? So we still have a lot to research because we have to understand how it works so that we can promote physical exercise general population. Because according to WHO, 80% of people now have a sedentary lifestyle. They're not meeting the daily physical activity. Then also we can identify these physical mimetic drugs. So exercise actually is a polypill because different organs actually respond to exercise. I will briefly talk about our findings with the we discovered adiponectin from fat cell and apelin from muscle as mediators, and also the biomarker in the blood for clinical research. So muscle-specific apelin actually is linked to sarcopenia, and it can increase by physical exercise to counteract sarcopenia in aged mice and humans. So in animal, what we can do is we can overexpress apelin in the muscle by injecting the adeno-associated virus to overexpress the apelin in the muscle. What we observe is after like 21 days, 3 weeks time, so increasing apelin can decrease the depression-like behavior in the behavioral test, like decreasing immobility time and also increased grooming in the school stress test. While in the transgenic lockout mice, we lock out the apelin muscles. So what we see is after running, these mice without muscle apelin, they didn't show any decrease in depression-like behavior. So indicating that muscle apelin actually is required for antidepressant effect of physical exercise. So in short, so what we found is with a physical exercise, it can increase muscle apelin secretion and go to the hippocampus and activate its receptor APJ in the hippocampus to increase AMD receptor function and increase hippocampal synaptic and structural plasticity and leading to antidepressant treatment effects. So suggesting that enhancing apelin signaling actually can elicit antidepressant effects. So any apelin receptor agonist like activating APJ could be a potential antidepressant. And in addition, we found that the fat cell secreted adiponectin also enhance newborn cell in the hippocampus and then elicit antidepressant effect. And recently we found that physical exercise increase adiponectin and also reduce tau phosphorylation by enhancing adiponectin and PP2A pathway. So suggesting a potential pathway where regular exercise can reduce the risk of Alzheimer's disease. And not only chronic or continuous exercise, single ball exercise in terms of like 30-minute treadmill running also have the beneficial effect in reducing mood disturbance in university students. So we can see that after 30-minute exercise, there's a significant reduction in the mood disturbance in the university students. So by building an animal model, we replicate this in the animal. What we see is the single ball exercise in terms of 30-minute treadmill running can increase the calcium influx into the prefrontal cortex, particularly in the glutamatergic neurons in the medial prefrontal cortex. And also with the two-fold imaging in the prefrontal cortex, the anterior cingulate cortex subregion, we observe increase in spine formation rapidly in the medial prefrontal cortex after exercise. While if we're blocking adaptor protein called APOL1, which is downstream to adiponectin, it can abolish the exercise effect. So entire study revealed that if your single ball exercise, 30-minute treadmill, can elicit antidepressant effect because of increase in adiponectin, which can activate the glutamatergic neurons in the anterior cingulate cortex, and then triggering downstream neural pathway for spine formation. And this spine formation actually strengthening the neural circuit connection in the ACC2 and anterior dorsal thalamus, so which is a very novel neural circuit in response to syncopal exercise. And then we discover exercise mimetic drugs with a receptor, adiponectin receptor agonist called adipiron, which can bind to receptor 1 and receptor 2. So we compare exercise effect and adipiron treatment. What we found is in diabetic animals, so adipiron treatment and exercise have a comparable effect in increasing newborn cell in the hippocampus, indicating that adiponectin receptor agonist actually can elicit similar neuroprotective effect to physical exercise. So now what we're doing is considering safety and also less side effect of traditional Chinese medicine. We are working on the traditional Chinese formula and also some herb-derived small molecule with adiponectin enhancing effect as a rapid and sustained antidepressant treatment. And also we discover the active ingredient extract from a long-term use of traditional Chinese medicine called yuezhi pill has a strong antidepressant effect. So for traditional Chinese medicine, there's a different herb in the formula. So in the yuezhi pill, so guardian fruit is the major herb, and we identify 2 active ingredients extract from this major herb, and we patent this drug composition because of a strong anti-inflammatory and neuroprotective effect. And what we found is the animal model with depression, so treating with these 2 active ingredients actually elicit antidepressant effect and by promoting hippocampal neurogenesis and synaptic plasticity. And recently, we also work on the air pollutant issues. Air pollutant induced cognitive impairment in general population because the air pollutant is a global issue nowadays. So we cannot escape from the polluted air. But what we found is in daily drinking water with the active ingredient extract from blueberry and also resveratrol, which is a very strong antioxidants, so it can restore the cognitive impairment induced by PM 2.5 in animal model. So yeah, that's all for my presentation, showing you our current work. Thank you.
Thank you very much, Sonata. You have done a lot of work. We should give you half an hour to do the presentation. Sorry. And now, the last but not the least, I would like to invite Professor Bolton Chow, also from PolyU Mental Health Research Center, to present his work. Bolton, please.
Thank you, Professor Chan. And I suppose I have shared my screen now, and hopefully everybody can see my slide. I'm glad to be here and to take this opportunity to very briefly share with you about my work. So I've put up this topic because I think the psychological process of reinforcement learning is important to everyone, but not just that is important to us human, it's also important to animals or even artificial intelligence. But here in my talk, I focus on cross-species comparison and to cover some work that we have done using neuroimaging and neurostimulation. So imagine if you make a costly mistake at work. What your brain actually has to do is to, well, apart from being very stressful, then you need to figure out how I can do it better next time. So this is exactly an example of reinforcement learning, which is the core system behind behavioral changes. And there are many examples of behavior changes that we have in life, such as quitting bad habits. For example, reinforcement learning can also be disrupted in many psychiatric disorders. And as I just mentioned, reinforcement learning is also a very important part of many AI algorithms. But how exactly does the brain implement this? So here I'm going to share my cross-species research using this technology I just mentioned. I'll focus on the comparison between humans and monkeys, both of which are primate species. So let's first talk about a part of the brain called the anterior cingulate cortex, which Professor Sonata Yao also has just mentioned. So it is a brain region that is remarkably similar in monkeys and humans, and also rodents, as Sonata has just mentioned. So in our 2019 study, we had monkeys performing a reinforcement learning task while brain activity was being recorded using fMRI. So interestingly, we saw activity in the anterior cingulate cortex that was related to information about behavioral change. So this precise part is shown here in blue, if you can see my pointer. And then by using non-invasive brain stimulation, we show the causal relationship. How we did that is that we temporarily disrupted the anterior cingulate cortex using a technique called transcranial ultrasound stimulation. And then the consequence is that the monkey showed reduced ability to implement these adaptive behavioral change. So they are poorer after the ACC was disrupted at when they needed to make behavioral changes. So recently we ran a very similar study with human participants. And again, we repeated the same procedures, asking human participants to go through this fMRI procedure. And then we replicated the signal in the anterior cingulate cortex, here highlighted in red in color. So we also recently have data involving human participants having the ACC stimulated using ultrasound, but this time we used an excitatory protocol instead of inhibitory protocol. The exact finding hasn't been confirmed yet, but our preliminary results suggest that these people with the ACC excited, they also made more adaptive behavior changes. So there's one extra piece of information that I would like to share with you. While the anterior cingulate cortex is largely similar across primate species, our recent human fMRI data reveal something extra. Here we also see very strong involvement of the frontal polar cortex during behavioral change. And interestingly, that this finding can only be seen in the human brain, but not the monkey brain. So the exact role of the frontal polar cortex in reinforcement learning is still not fully clear, but at least recent anatomical findings suggest that, well, the frontal polar cortex that I'm labeling here in red is a region unique to the human brain. And a better understanding of this function may help us to explain why humans can learn faster and handle far more complex situations than other primates. So my recent review paper published in Brain discusses the broader and more complex function of this fascinating brain region. I haven't got much time to share with you about this view, but if you are interested in it, I encourage you to read this paper. Thank you.
Thank you very much, Bolton. Again, this is wonderful work. Now we'll move to the panel discussion session. George, floor is yours.
Yes, thank you very much. Wonderful presentations. Many thanks to all speakers for inspiring us. We're ready now to take your questions. We'll go through the questions, and I'll be happy to moderate and send them on their way. This will be the moderation will be done in collaboration with Professor Zhang Weixiong.
Okay. We have a few questions from the floor. The first one is, thanks for this inspiring presentation. I have a very interesting question. Does it means that it could seize every moment? No, the question referred to your experiment, putting implant into the brain, and then you try to get the words out of the thinking. So does it mean that it could seize every moment of brain activity? Actually, not every motion word in our mind is what we would like to convey or transmit to the surface. So in other words, you capture
yeah.
the signal, it may not be that person's intention to really, get out.
How do we ensure
Yeah.
privacy?
Exactly. I think that's a very, very important question. So if I can just restate the question is, how do we make sure that what we're decoding from the brain is what someone actually wants to say versus their inner thoughts? It's a very important question. And the decoder that we use primarily is decoding what people are trying to say, because it's decoding the different movements and intended movements of the vocal tract. So for now, this technology is primarily about trying to restore the movements that are lost, not directly translating the thoughts. In the future, more and more effort is going to be looking at what those inner thoughts are and having to build in some mechanism that you only transmit the ideas that you want to express, not just the ones that you're thinking about. This will be important. I think this is a very difficult thing to decode. That being said, because that kind of information of our thoughts is very distributed. It's not just one area that is encoding that information. Thank you.
We have another question. It's very long because he asked 3, but I'm going to pick just one. This is someone from Fudan University. He mentioned that he is from a lab of your former student. Let me see if I can bring that up. From Dr. Wu Jing Song, if you look on that name. Okay, let me answer the second question he asked. Second, most speech BCI studies today focus on decoding from VSMC. Besides VSMC, what other brain areas do you think could be useful for speech decoding in the future for patients with aphasia caused by stroke involving VSMC and even large brain area? How far are we from being able to decode speech in these patients? That's a little bit technical question.
So the question is really about how do we go beyond the speech motor cortex, the area that I was just describing as a motor commands. And for many people, for example, that area could actually have a stroke itself. So you can't decode from it. And so we need to look for other areas, particularly for people that have language disorder, for things like not necessarily a speech disorder, but language disorder called aphasia, where you have injury to Broca's or Wernicke's area. And so I think we need to think carefully about, how semantic information is encoded in the brain. Like I said before, this is a difficult problem because the information is very distributed, whereas in the motor system, everything is become very compact and modular. So we may need new kinds of technology that are recording from many cells in a distributed way in order to move beyond speech motor cortex. Thank you.
Okay, another interesting question. Thanks for amazing presentations. I have a question for Professor Chang. I'm wondering if we would be able to use the same system for decoding animal speech, even if we don't understand it yet. So in other words, my understanding of the question is, we don't know even the vocal system, or not a vocal system, the alphabet of the animal. So how can we go apply your technique to understand animal communication?
Yeah, it's a good question. So the technique that we developed is actually very general technique. And the input is the neural activity. The output is the speech or behavior that we're trying to decode. And the modern machine learning, I think, is very, very good at solving this kind of problem, provided you have enough data. So I do think that you can use the same algorithm actually to decode communication from other animals. If your question is about how to communicate and translate the communication signals from other animals, you don't necessarily need a brain-machine interface to do that. You can basically use the same kind of models to try to understand what's the nature of the whale call or the bird song using what we call self-supervised learning. Try to understand what the structure is. And how to translate it to English, I think it's going to take a lot more work, but it's a super interesting area to see if that can be done. Thank you.
We also collect some questions, when audience that register for this session. So one of them is the following. How does the brain work while we sleep? And what's the mechanism behind seeing a dream or having a dream, as well as talking during dreaming? I wonder if you have done any, related research along this line.
We are interested in understanding this. Right now, a lot of the recordings we do are during the day, but with the new kind of devices that we're developing are going to be fully wireless. We can record through the day and night and try to understand how learning occurs in sleep. A lot of the research now suggests that the consolidation of even movement and learning, for example, when someone is learning to control the robot with the brain activity, that consolidation actually happens during the sleep period. And so there's certain periods of sleep, in particular the REM period, where basically you can be in a dream state. And when you're speaking, basically it means that you're deep into a dream state and interacting with others. We should be able to tap into that. We should be able to decode that. Something that I don't think has ever been done before, but it will be very interesting to see if you could decode essentially what someone is actually thinking about during REM sleep. Thank you.
Okay, another question from the earlier registration period. Given the role of neuron plasticity in recovery, when patients regain speech through neural the therapy you conducted, do you observe functional or structural change in other brain regions? Are there any potential risk or unintended consequence associated with this neuron reorganization?
It's a good question about the plasticity. And I think one thing that we're interesting to understand is how much the brain is changing to adapt to the brain-computer interface. We don't fully know. I can tell you that it definitely does when people learn to do this task, and especially when they become very proficient. It's definitely related to learning, and that's a form of plasticity. Just thinking about how the brain is using its activity to control something that never existed, for example, a robotic arm. And so some of it relies on thinking about how your own arm moves, but a lot of it actually is adapting to a new limb, for example, that you never had. And so we believe in what we've seen is that learning is actually quite important. In fact, we call this co-learning, where the BCI is essentially learning to decode the neural activity weights, but the human in the brain is actually trying to understand how the BCI decoder is working. Both are trying to optimize towards the best performance. And so this is a very active area of research. I think it's just starting to begin. But any way that we can accelerate the development of co-learning, I think will help us achieve the goals much quicker. Thank you.
Here's another interesting question. How stroke affect the functional organization of speech? in which brain regions?
Well, there are different parts of the brain control both language and speech. And language I would refer to as sort of like the syntax, the grammar, the meaning of the words. And the speech, in this particular talk, I'm referring to the production of the sounds and the motor commands, the movements. And so there are certain areas that if you had a stroke, for example, in the area that we study for BCI decoding, that's the ventral sensorimotor cortex. It's the lateral ventralmost part of the motor cortex. A stroke there causes facial weakness and also causes dysarthria. So people know what they want to say and the words are there, but they cannot be articulated fluently. Either it's slurred or unintelligible. The other condition is what we call aphasia, which is the areas that are generating the concepts of the words and the form of the words. This includes the temporal lobe, different parts of the parietal and also frontal lobes. It starts to become distributed very quickly. So it really depends what part of the brain, whether it's part of the speech processing network or the greater language network.
There's another question from the floor. Thank you for all your wonderful talks. Can I ask Professor Chang, when decoding internal monologue, how does the system distinguish between active linguistic intent and passive cross-linguistic interference? For example, the L1 structure intruding on an L2 target.
So I think there are two questions there, but the first question is about decoding actual speech versus the inner speech. And I think we talked about that a bit earlier, that what we're really decoding is what is the intended motor commands to the vocal tract. So what people are just thinking about or inner speech doesn't really get translated. And one of the findings that we had is that inner speech is not necessarily the same part of the brain even. Some people would argue that, that there is some evidence of inner speech, but for the most part, it's not very well encoded there. And on top of that, if people have injuries, for example, in that part of the brain, the sensorimotor cortex, they still have a perfectly good ability for inner speech, even though they cannot, fluently articulate. So the second question was, cross-linguistic interference. And there's definitely a possibility of that if you speak two different languages. Everything that I showed you actually relies on having a very specific model of language. We use the statistics of English to help increase the probability of the correct answers and the speed. And you need to have a model that is, sort of accurate for the statistics of a given language. Otherwise, it won't work, at least not in the current versions.
Great, thanks. We still have a few questions for Professor Chang, but I'd like to move on a little bit, to see if we still have time at the end. We will come back. I want to cover, the other panelists. Here's one question I want to ask to all of you. What are the most important technology advancement these days? For example, possibly new imaging technology, brain stimulation techniques, and another direction it's moving really fast is a single cell spatial chromotomic profiling. So would you like to see in the near future, what kind of new technology that you may be able to use, and could such advance change your research direction or affect your work? This is, looking forward.
And let me butt in here to say that this should be a discussion. So let's
Yes,
try
yes.
to share opinions here on this.
So
Maybe I should point at people and ask them to chime in.
I can chime in. I think for me, one of the biggest advancements currently is actually in the miniaturization of most technology, and in particularly actually the adoption of wearable technology. We're going to get to a point, which we kind of already are at, but very, very soon, where we can have access to so much personal and wearable data from so many individuals who are struggling with neurological conditions that, quite frankly, has not been available at any point of time in human history. And what I think is exciting to me, and I think what will change research the most, is that we can really start to understand how different countries, different upbringings, different situations can actually impact or affect treatments. And we can hopefully flatten this line that has been seen where most research is done in one specific region or one specific field. I think it's an incredibly exciting time. I think there's going to be so much to learn. So that's my kind of 2 cents.
Thank you, Jonathan. Any other opinions on this question?
If I may kind of add on to
Yeah.
what Jonathan have mentioned, I definitely agree. I think there's such an exciting time that we are able to use hardware and software and platform data analysis to be able to understand our brain more. And I definitely think it is giving us an opportunity of this understanding of how our brain is working far more better than we were able to before. So I think there's a lot of interest there. On top of that, I think one of the really fascinating area is the non-invasive ways of being able to support our brain health in total. I think there's a lot of more understanding of how external stimuli impact our brain and how we process information. So that's definitely one of the key areas that I'm interested in.
Great.
Yeah.
Anyone else?
Yeah.
Sonata, I see you unmuted yourself.
Yeah, I'm totally agree with the technological advancement with the human brain computer interface, and also the brain stimulation for the treatment for psychiatric disorders, for different kind of neurological disorders now emerging, which can help with the patient who cannot take medication. Those are very important advancement for human health, and also so because I'm an animal work, biological work, so I think we should also have done a lot of work on the spatial single cell spatial transcriptomic analysis, which give us some more detailed information about how disease change the single cell gene expression or protein expression in a specific brain region, and what type of neurons actually are being modulated. So we can have more precision medicine for treating neurological disorders, particularly for the psychiatric disorder that I have been working with Joan. So those are technology very important for precision medicine in the coming future.
Thank you. I'm interested in hearing from Bolton as well.
Yeah, thank you. I can see that, there are 2 sides of technology that is going to be moving very fast in the coming years. One is to read neuroactivity, as Professor Chang mentioned. There are these BCI interfaces that has been developed so rapidly, and there are, invasive solutions like Professor Chang's inserting, electrodes, not directly into the brain, but on the surface of the brain. There are other non-invasive technologies. So that would be one camp that is going to be definitely moving very fast. I think all of us have heard of, Teslas or Neuralink's development these days. They may not be the best, but they're probably one of the, most eye-catching developments. The other camp definitely would be, different ways of disrupting the brain or stimulating the brain. And in the past, we cognitive neuroscientists struggled to how we can stimulate deeper brain regions. And recently, we've got these new ultrasound techniques that allow us to reach deeper into the brain. So that is, definitely will be happening. But I guess if you ask me what would be the greatest challenge, it's going to be some more philosophical issues that we are facing. For example, privacy issues. If everyone is trying to, implant, non-invasively or invasively these BCI devices, So how's the world going to handle this? How are we going to protect privacy of people? I think it's a very similar issue these days AIs have brought to us. And I guess, well, my stance is that the technology is neutral. How we human can make good use of it is a different question.
Thank you. Last but not least, Professor Chang, what is your opinion on this? You have pioneered the development of bespoke technology for your work. What other technological developments excite you these days?
Well, I do think that we're going to, in the coming years, see a proliferation across really all different dimensions of invasive and non-invasive. I think at a philosophical level as well, I think that it's even less about the hardware, more about a bigger question about, where is the line between what is human and artificial? I think we're starting to see this challenge with artificial intelligence, but when people are directly linked to the artificial intelligence directly through the brain, I think this is going to pose some very, very interesting and challenging questions to grapple with. Today, I showed you some examples of using AI to essentially interpret, someone's speech and thoughts. That's already an example of a merge between, natural biology coding and also artificial intelligence. I think that as time goes on, we're going to start to see more and more of that. And the line between what is human and what is not is actually going to become very blurred. So we need to be aware of that and think through what the implications are.
Yeah,
sorry, George, let me jump onto this question regarding the AI. I have a question for both Professor Chen and Bolton, because in your research, more or less, you touch upon AI, both in reinforcement learning and in Professor Chen's work. You also use a neural network, all those things, you try to decode. Although, you did this more than 10 years ago, starting from there, you actually used like a generative AI technique. It's a way before ChatGPT, in my view. So my question here is, how do you think, because, AI these days, neural network, it's originally start from neuroscience. We actually get the clue from neuroscience. So what do you think for your work? I really see, that there's a good potential you can feed into teach human how the brain work. So how can your work help AI research to move forward? Because AGI is a big topic on the table. A lot of billions of dollars these days on Wall Street. So what do you think? I want to poke your brain, see how you see this.
Well, maybe I'll go first, and I'm sure that Professor Chan has more brilliant thoughts about it. I guess, one obvious issue nowadays with AI is energy problem, as we can see from the news. It is it's a very energy hungry technique, which in contrast, our brain is highly efficient. Imagine how much energy is required to keep our brain alive, as opposed to how much energy you need to feed AI. So one clear direction, I'm sure that those AI people are working on that, is to how we can leverage the efficiency in the brain and use these algorithms that we might implement in the brain that is highly efficient and put that into AI algorithms. And it might be the case that it might not do all the good works, as in there's always a cost. For example, human behavior is not always rational, or it's not always optimum. For example, if people are stressed, they might perform poorly. But in general, our brain is designed in a way that in order to allow us to be efficient overall. So maybe that would have some insight for AI development in the future if we can put in some brain principles into the AI algorithms.
The only thing I would potentially add is that I think we're going to see a new kind of neuromorphic computing that's no longer just brain inspired, but brain trained. Because I think that what happens in the coming years is the data that we have from neuroscience is going to scale. So it will no longer be about just the basic principles that we're learning from the brain, but actually the brain data itself will be used to train new forms of AI. So this is a very interesting direction. And I'm curious to see actually how much you can accelerate computation, both at the hardware and software level.
Maybe last question here. So we have two panelists. They have experience, or actually three, including Professor Chen. Two of you, Lucy and Jonathan, they start your own business. So the question here is, how do you connect the dots from basic research, technology, all the way to patient care? So it's very much, you go to almost, go directly to patient. How do you make the initial dots? Because this is also, I bet a lot of audience would like to ask, right? Start your own business from discovery.
I'm happy to go first in case it's helpful. So I've been developing, so this is my second neurotechnology company. And I think it's such an important question about, I used to do the research. So the previous research where it was looking to paralysis or multiple sclerosis or insensitivity to pain, it was very, very quite research-based. So it was very much more conceptual, and we were looking to do very fundamental science of it. But through my kind of experience of actually being a patient, of going through the brain surgery myself and going through quite a lot of challenges through that, really kind of inspired me of how it's very, very important for us to go from the research to actual impact. So unfortunately, of course, we would want to cure all neurological conditions, but what we wanted to really do as myself and our team is being able to translate what we currently are aware of, such as things as what Jonathan is doing, which is incredible that there has been lots of studies around curing in Parkinson's, but actually the path of translating is really, really difficult. That's why I believe that you brought this question is, I think it's very, very important to talk with the stakeholders, including patients, carers, hospital systems, and regulators, and so on, of what is achievable right now and what can we actually do in the future, and understanding that pipeline, really understanding the current careway pathway has been really, really important point, so that we can make sure that what we're developing can be actually handed over to people who need it the most. So it's a big question of how do we translate from the research all the way to the impact. There's a lot of stakeholders, and I do have to say Cambridge NeuroWorks has been really impactful for our journey, because it did understand the ecosystem that is required to bring the science to actually impact real world. So I think it's really, really important to get that stakeholders, whether it's patients, clinicians, hospital, everyone involved, so that you can understand to fill in the gap.
Thank you. Jonathan? Jonathan?
I think for me, probably the most important thing is to really understand the core value propositions of actually what you're selling. And oftentimes it's very difficult, because what you think is what people want is actually completely opposite of what people want. I think academia is fantastic actually for translating concepts and for building things and de-risking it. I think the hardest part in business is actually taking what you know is works and wrapping it up into a certain package that actually people are willing to pay something for. And a lot of the time you can only do it once. And if you don't do it well once, it's hard to do it twice. So it's really understanding what are those core features? What is the 20% of features that you can include that provides 80% of the impact? And really just executing those 20% exceptionally well. I think that's the core challenge, and focusing on just that one thing at the start of the journey.
Thank you. Professor Chen, do you have anything to add? Because you already also have your own company,
Yeah, we have spun out a company, and I really like what Lucy and Jonathan had to add, because it's so personally driven, because they really understand where the needs are. For me, the intention was never about starting a company. I don't consider myself as an entrepreneur in any way. But I think the reality that I myself encountered was that we basically couldn't go any further without new technology. And I realized we had to create our own new technology. And there's a chicken and egg, because what I showed you was kind of like what many people would call minimal viable product. It was like essentially tremendous de-risking of high, something that was not even clear if it would work. And then over a couple of years after demonstrating that it had some promise, I came to the conclusion that I need better technology. I need something fully wireless. We need more channels. I can't just be gluing these things together anymore. And so it was partly out of necessity to continue to develop the technology and to make impact. This was not really something that was born from an idea. It was really born from seeing it work and then thinking carefully about what would be required in order for it to be sustainable as a business. And that's where we're thinking. And I think it's a very, very exciting time. I love how your center is really thinking forward about how to translate research into commercial opportunities. I think that's a very recent trend, because historically, at least, my university and many others, that was not exactly, how people thought about academic career. But now I think it's very, very positive to see encouragement and to see if we can make some of these things really make impact in the clinical field. So I think the short answer is I would like to just see the technology that we have move into the real world, as opposed to just papers and talks and demonstrations. Ultimately, that's I think, driving all of us. And so, one of the key ways, of course, to do that is to build a business that is going to be sustainable and generate value to patients. And I think that's going to be really key.
Thank you. Thank you. Thanks for all the answers and the discussions. So due to the time constraint, let me get back to George. George.
Thank you all again. Yes, thank you for sticking with us. For some of you, it's very late. For some of you, it's time to go to work and operate. Thank you all. Thanks to the presenters. Thanks to the organizing committee. This has been a fantastic experience working between two institutions, with a lot of buy-in from both institutions and a lot of support. This will hopefully be the first in a series of 3. Please stay tuned for more information about the follow-up events. And please visit the websites of both institutions and subscribe to the different media venues to get information. For example, from Cambridge NeuroWorks, Connect Health Tech is the application that would remind you of upcoming events. So with that, thank you very much on behalf of Cambridge NeuroWorks. Professor Zhang, back to you.
All right. So I think this will pretty much conclude our the first seminar on this series. And I hope everyone will keep an eye on our announcement. We should have more to come. Thank you.
Thank you all.